Okay, let's unpack this. We are diving into probably the most foundational molecules of life, proteins. And this isn't, just basic biology 101. This is really molecular architecture we're talking about.
Absolutely. It's the blueprint and the building blocks all rolled into one.
Yeah, exactly. And the stack of material we looked through, it gives this incredibly deep, comprehensive roadmap for these amazing structures. It takes you right from the tiniest chemical bits all the way up to their final functional 3D shapes.
It really covers the ground.
It does. So our mission today is really to give you a bit of a shortcut, right? A high-level view of exactly how these molecules, which, let's be clear, form the basis of structure and function in pretty much every living system, how they're put together, how they stay stable, and ultimately how they behave.
And the scale is just immense when you look at it.
Oh, totally. I mean, the raw statistics are staggering. Proteins are easily the most abundant organic molecules in any living system. They make up something like 50% of the cellular dry weight. That's huge.
Half the weight. When you take out the water, it tells you how central they are.
And they're needed everywhere. The sources break down their roles into two really broad categories. First, you've got the static or structural roles. This is the physical stuff.
The scaffolding.
Exactly, the scaffolding. Think of it like the brick and mortar of the body. You've got collagen in your tendons, your skin, keratin making up hair and nails. It's all about providing strength and form.
Solid, dependable structures, but then you have the other side of the coin.
Right, the dynamic roles. And arguably, these are maybe the most crucial for just keeping things running day to day, wouldn't you say?
Well, absolutely. These are the functional heavy lifters.
Yeah.
The real working horses inside the cell. They're acting as enzymes, speeding up basically every essential chemical reaction.
Millions of times faster sometimes.
Unbelievably faster. They're hormones carrying vital signals all over the body. Immunoglobulins for defense against invaders. They're key players in muscle contraction, genetic control. It's the cell's operational staff, really.
And the efficiency of life, it just boils down to how precisely these proteins function.
It does.
And here's what I find truly elegant. Every single one of these massive, complex, multitasking structures, whether it's that structural collagen or a super-specific catalytic enzyme, is built from the exact same simple foundation.
20 building blocks.
Right. They are all polymers, these long chains built exclusively from alpha amino acids. So if the finished protein is like some kind of sophisticated, custom-designed building...
Then the amino acids are the essential bricks. Chemically versatile bricks, mind you.
Exactly. Versatile bricks.
So to really get into this deep dive, we absolutely have to start with the brick itself. Understand the amino acid first. Okay, so let's look at the core structure of an amino acid. Architecturally, it's simple but, well, profound in its implications. Everything centers around one specific carbon atom.
The alpha carbon, right, the alpha carbon.
That's the one. The alpha carbon, it's the central anchor. And it has to be attached to 4 different things, typically. You've got the basic amino group that's made from an H2X, and the acidic carboxyl group made from COACs.
Okay, basic and acidic. And critically, there's the side chain. That's the R group, isn't it?
Exactly, the R group. And this is the game changer. It's the identifier, the chemical signature that dictates pretty much everything about that specific amino acid. Its size, its charge, whether it likes water or hates it.
It's personality almost.
It's chemical personality, absolutely. And it dictates where it's likely to end up when the final protein folds up. It's what makes each of the 20 standard amino acids unique. Hashtag 1.1, the general structure and ionization. And they're called alpha amino acids specifically because both of those defining groups, the amino and the carboxyl, are bound to that central alpha carbon. But here's where it gets really interesting. How they actually exist in a biological system, like in our cells.
Right, because they're not usually just floating around neutral, are they?
Almost never. Not in that simple textbook neutral form. In the biological system, especially around physiological manthrem, which is near neutral, well, as an acidic carboxyl group readily gives up a proton. It becomes negatively charged.
Okay. Loses a proton.
And the basic amino group, it picks one up, it becomes positively charged, methymin H3 plus CO. So the resulting molecule is what we call a zwitterion, or dipolar ion.
Zwitterion, sounds fancy. So it's got both a positive and a negative charge at the same time.
Exactly, simultaneously. Which means even though it has charges, the molecule overall is electrically neutral. And this perfect balance happens only at a very specific pH value for each amino acid.
Which is its isoelectric pH, or pI.
Precisely, it's pI.
Oh.
And this dual nature, this ability to carry charge but be neutral overall, is why amino acids are called ampholytes. They can act as both weak acids and weak bases.
Ah, so that's key for buffering, right? Resisting pH changes.
Absolutely fundamental for their buffering capacity and how they interact in solution. It's critical.
Okay, and this structure also relates to their shape in 3D.
It does, directly.
Yeah.
Remember, the alpha carbon is bound to 4 different groups. You've got the amino group, the carboxyl group, a hydrogen atom, and that unique R group.
Unless it's glycine.
Right, good point. Glycine is the exception. Its R group is just another hydrogen atom. So the alpha carbon in glycine is attached to only 3 different types of groups. But for all the others,
19 others.
the other 19, the alpha carbon is bound to 4 distinct groups. That makes the alpha carbon asymmetric, or chiral. And this asymmetry leads to what we call optical isomerism. They can exist in 2 mirror image forms,
L form and D form.
L and D isomers. And when it comes to life as we know it, our bodies are incredibly selective. Proteins in basically all living systems are made up exclusively of the alpha amino acids.
Only the L form. What about the D form? Does it just not exist?
Oh, D-amino acids definitely exist in nature, but they're usually found in more specialized places, like in the cell walls of certain bacteria or in some antibiotics produced by microbes. But for the core machinery of life, the proteins doing all the work, it's strictly L-amino acids. It highlights this really strict chiral purity that life seems to require. Tag 1.2 structural classification. The 7 families.
Okay, so we've got these 20 versatile bricks. Now, to make sense of how a long chain of them might fold up, we need to organize them somehow. The source material groups them based on the chemical nature of that R group, right? Into 7 families.
Yes, 7 distinct structural categories. It's a useful way to think about them. We can start with the simplest, the ones with aliphatic side chains.
Aliphatic meaning just carbon and hydrogen, basically. Non-aromatic.
Pretty much. Simple hydrocarbon-based groups.
Yeah.
This includes the smallest ones, glycine and alanine. And it also includes the important branched chain amino acids, or BCAAs.
Oh, the BCAAs. Valine, leucine, isoleucine, you hear about those in supplements?
You do. They're highly hydrophobic water-hating, and they play really critical roles, especially in things like muscle metabolism and cell signaling.
Okay, family 1, aliphatic. What's next?
Next, we have amino acids containing an oxygen group, specifically a hydroxyl group. These are the hydroxyl group containing amino acids. That's serine and threonine.
Serine and threonine. And that hydroxyl group, the Mothram A's, that makes them polar.
Highly polar, yes.
Hmm.
Excellent sites for forming hydrogen bonds, which are crucial for structure. And importantly, they're key sites for post-translational modifications. Things like phosphorylation, adding a phosphate group, often happen right there on that hydroxyl group.
Which can act like an on-off switch for the protein's activity.
Exactly, a major regulatory mechanism. Okay, third family, the sulfur-containing amino acids. 2 main players here.
Cysteine and methionine.
Right. Cysteine is unique because it has a reactive sulfhydryl group. Now, this group can covalently bond with the sulfhydryl group of another cysteine molecule.
Forming cysteine.
Forming cysteine, yes, via a disulfide bond. This is a very strong covalent bond, and it's one of the most powerful forces for locking a protein's 3D structure into place.
Okay, so disulfide bonds are like structural staples. What about methionine?
Methionine contains a thioether group, methymet CH3, instead of the sulfhydryl. It's less reactive in that sense, but it's critically important because it almost always serves as the starting amino acid when a protein is being synthesized in the cell.
The start signal for translation. Got it. Sulfur done. What's family 4?
Moving on to the ones that carry a charge at neutral pH. First, the acidic amino acids and their amides. The acidic ones are aspartic acid and glutamic acid.
Sometimes called aspartate and glutamate?
Yes, because at physiological pH, they've lost a proton from their second carboxyl group in the R chain, so they carry a net negative charge.
Okay, acidic means negative charge, and they're amides.
Their amide derivatives are asparagine, from aspartic acid, and glutamine, from glutamic acid. The extra carboxyl group is converted to an amide group, methylCOMH2Y. This neutralizes the negative charge, but they remain very polar molecules, good for hydrogen bonding.
Okay, acidic and amides. Next must be
The counterparts. The basic amino acids. These are highly basic, meaning they readily accept protons, so they carry a net positive charge in the cell.
So, acidic or negative, basic or positive. Who's in this group?
This group includes lysine, which has a long side chain ending in an epsilon amino group. Then there's arginine, which boasts the guanidino group that's a very stable, highly basic group. And finally, histidine with its imidazole ring.
Histidine's interesting, isn't it? Because its charge can change around physiological pH.
It is. Its pKa is close to neutral, making it really versatile in enzyme active sites, acting as a proton donor or acceptor. But generally, these positive and negative R groups are essential for forming those ionic interactions, those salt bridges, that help stabilize the final 3D architecture.
Like little magnets holding parts of the chain together. Okay, 2 families left.
We then hit the ones with ring structures.
Yeah.
The aromatic amino acids. These are phenylalanine, tyrosine, and tryptophan.
Big, bulky rings.
Yes, and quite hydrophobic, generally.
Hmm.
They all contain these stable ring structures. Phenylalanine has a simple benzene ring. Tyrosine is similar, but with an added hydroxyl group, making it a bit more polar. And tryptophan has a complex double ring structure called an indole ring.
And these are the ones that absorb UV light, right? That's how we measure protein concentration in the lab.
Exactly. Their aromatic wings strongly absorb UV light around 280 nanometers. Very useful property. Okay, last one.
The odd one out.
The structural oddity, yes, the amino acid, proline. It's unique. Its side chain isn't just hanging off. It actually loops back and bonds covalently to the backbone nitrogen atom itself.
Forming that rigid ring.
A rigid pyrrolidine ring, yes. This means that instead of a standard primary amino group, meth or MNH2 like all the others, proline has a secondary imino group. And that rigid ring structure severely restricts rotation around the peptide bond it forms.
So it acts like a kink in the chain.
Precisely. It often introduces sharp turns or kinks in the polypeptide backbone. It can't fit easily into regular structures like the alpha helix, so it often acts as a helix breaker or a turn inducer. Very important for dictating the final fold. So, now that we have a feel for the structures, we can classify them in more functional ways. And probably the most important classification for predicting how a protein will fold is based on polarity.
Right, because proteins usually fold up in water inside the cell, so how the amino acids interact with water is key.
Absolutely key. Polarity dictates where an amino acid is likely to end up in the final 3D structure. So first group, nonpolar hydrophobic.
Water-hating.
Water-hating, no charge. This includes alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan, and also proline. Because they dislike water, they tend to aggressively cluster together in the interior of the protein.
To hide from the water, like an oil droplet forming.
Exactly. This hydrophobic collapse is often considered the primary driving force behind protein folding. Get the oily bits away from the water.
Okay. Hydrophobic core. Then what?
Then we have the polar no-charge amino acids. This includes glycine, serine, threonine, cysteine, glutamine, asparagine, and tyrosine. They have groups like hydroxyls, the sulfhydryl, or amide groups. These don't carry a net charge at neutral pH, but they are polar.
Meaning they like water?
They're hydrophilic, yes. They can form hydrogen bonds with water and also with each other. So you tend to find these on the surface of the protein, interacting with the cellular environment, or involved in forming the hydrogen bonds that stabilize secondary structures.
Okay, nonpolar inside, polar outside are bonding. And the charged ones.
Right, the charged ones. We have polar positive R group. These are the basic amino acids, lysine, arginine, and histidine. And polar negative R group. These are the acidic amino acids, aspartic acid and glutamic acid.
The bases and acids.
Exactly. These are usually found on the protein surface too, interacting with water or forming those crucial ionic interactions, the salt bridges with each other, which help lock the tertiary structure into its final shape. They also define the protein's overall net charge at any given pH.
Makes sense. Polarity drives folding. What about other ways to classify them?
Another really important classification, especially from a health perspective, is based on nutritional requirement. Our bodies are amazing chemical factories, but they can't synthesize everything from scratch.
Some building blocks we just have to eat.
Exactly. These are the essential or indispensable amino acids. For humans, there are generally considered to be 10 that are needed for proper growth and maintenance, and these must come from our diet.
Okay, 10 essential ones. Which are they?
The source lists the core 8 as valine-leucine, isoleucine, the BCAAs again, lysine, methionine, phenylalanine, threonine, and tryptophan. But we usually also include arginine and histidine in that essential list, particularly for certain life stages.
Why only sometimes?
They're often called semi-essential. Healthy adults can typically synthesize enough arginine and histidine for their needs. However, growing children or individuals under certain metabolic stress or illness might not make enough, so for them, a dietary supply becomes essential.
Gotcha. So essential and semi-essential. The rest we can make.
The remaining 10 are non-essential or dispensable. This means our bodies have the metabolic pathways to synthesize them, usually from other common metabolites. Examples include glycine, alanine, serine, proline, glutamate.
Okay. Essential, non-essential. Diet matters. Is there one more classification?
Yes, one more functional view, based on their metabolic fate. When a body breaks down amino acids, what happens to their carbon skeletons? Where do those carbons end up?
After the nitrogen is removed for urea or other things.
Precisely. Some amino acids are glycogenic. This means their carbon skeletons can be converted into intermediates like pyruvate or oxaloacetate, which can then be used to synthesize glucose or glycogen through gluconeogenesis.
So they can become sugar, like alanine, methionine.
Many of them, yes.
Yeah.
Others are ketogenic. Their breakdown products like acetyl-CoA or acetoacetyl-CoA lead directly towards the synthesis of fatty acids or ketone bodies.
They can become fat or ketones. Who are the purely ketogenic ones?
Only two are exclusively ketogenic, leucine and lysine. All else.
Leucine and lysine. Only fat ketones. Are there any that do both?
Yes, there are 4 clever ones whose carbon skeletons can enter both pathways. They are both glycogenic and ketogenic. These are isoleucine, phenylalanine, tryptophan, and tyrosine.
The aromatics plus isoleucine, so they offer metabolic flexibility.
Incredible flexibility. Their carbons can be shunted towards sugar production or fat ketone production depending on the body's needs at the time. It really highlights how interconnected these metabolic pathways are. Hashtag tag 1.4 non-standard amino acids beyond the 20.
Okay, this is where, as the source says, it gets really interesting. Because biology, well, it doesn't always stick neatly to that list of 20 standard amino acids encoded by the main genetic code, does it?
Not at all. The system has evolved some fascinating exceptions and additions. We absolutely have to talk about the unique standard amino acids first.
Like the 21st and 22nd?
Exactly. Selenocysteine is often called the 21st amino acid. As the name suggests, it contains a selenium atom instead of the sulfur atom found in cysteine. It gets incorporated into a special class of proteins called selin proteins, which are often involved in antioxidant defense.
But how does it get put in? I thought there were only codons for 20.
That's what's so amazing. The genetic code used for selenocysteine is actually UGA. Now, UGA is normally a stop codon. It tells the ribosome to terminate protein synthesis.
So how does it insert an amino acid instead of stopping?
The cell has evolved a complex machinery involving specific RNA structures and protein factors that, under the right conditions, essentially overrides the stop signal at that UGA codon and directs the ribosome to insert selenocysteine instead. It's a remarkable feat of genetic recoding.
Wow. Repurposing a stop sign. And the 22nd?
The 22nd is pyrrolysine. This one is found mainly in certain methane-producing archaea and some bacteria. It uses the UAG stop codon, another stop signal, for its incorporation. Again, specialized machinery allows the cell to read UAG as insert pyrrolysine in the context of specific messenger RNAs.
So the cell is literally redefining its own genetic alphabet on the fly. That's incredible.
It really showcases the evolutionary plasticity of the genetic code. But beyond these genetically encoded exceptions, there are other non-standard amino acids crucial for biology.
Like ones that are modified after the protein is made?
Precisely. Derivatives in proteins, created by post-translational modification. These modifications often add crucial stability or function that the standard 20 can't provide alone.
The big examples are in collagen, right? That structural protein.
Absolutely. 4-hydroxyproline and 5-hydroxylysine. Normal proline and lysine residues within the newly synthesized collagen chains get hydroxylated, and a mathrm group is added.
And why is that important?
These hydroxyl groups are essential for forming the extra hydrogen bonds that stabilize the incredibly strong triple helix structure of collagen.
Right.
Without these modifications, the collagen molecule is weak and unstable.
Which is what happens in scurvy.
Exactly. Vitamin C is a required cofactor for the enzymes that perform this hydroxylation. No vitamin C, no stable collagen. That's why scurvy leads to bleeding gums. Core wound healing tissues literally start to fall apart.
A direct link from a vitamin to protein structure. Wow. Any other key derivatives?
Another important one is gamma-carboxyglutamic acid, cool N-italica. This is found in several plasma proteins involved in blood clotting, like prothrombin. Specific glutamic acid residues get an extra carboxyl group added.
And that extra negative charge does what?
It allows the protein to bind calcium ions, mathrm Ka2 plus Vbit, very effectively. This calcium binding is absolutely critical for anchoring these clotting factors to phospholipid surfaces at the site of injury, initiating the coagulation cascade. Some blood thinners, like warfarin, actually work by inhibiting the enzyme that makes GLALA residues.
Fascinating. Okay, so we have modified amino acids within proteins. What about amino acids that aren't in proteins at all?
Yes, the non-protein amino acids. These have the basic amino acid structure, but are never incorporated into polypeptide chains during translation. Instead, they function as important molecules in their own right.
Like intermediates in metabolic pathways.
Exactly. The source mentions ornithine and citrulline. These are key intermediates in the urea cycle, the pathway our body uses to safely dispose of toxic ammonia by converting it into urea. They cycle through the pathway but never end up in a finished protein.
Okay. Any others?
Then you have amino acid derivatives that act as hormones or neurotransmitters. Thyroxine, the main thyroid hormone that regulates metabolism, is synthesized from tyrosine.
And the classic example must be GABA.
Gamma-aminobutyric acid, GABA, yes. It's the primary inhibitory neurotransmitter in the brain, crucial for calming nerve activity, and it's synthesized directly from glutamic acid, one of the standard 20. So these examples really show that the basic amino acid structure is incredibly versatile, used for much more than just building proteins.
All right, so we have a solid understanding of the bricks now, these versatile amino acids. Let's look at how we actually lay the foundation for a protein, which brings us to the primary structure.
Primary structure. This is simply the specific linear sequence of those alpha amino acids as they're linked together to form the polypeptide backbone. It's the fundamental blueprint.
And when we say blueprint, we mean it's absolutely critical, right? This sequence dictates everything else.
It is non-negotiable. This unique sequence is determined directly by the organism's genes, encoded in the DNA. This linear order which amino acid follows, which dictates every single subsequent step of folding. It determines the protein's final 3D shape and therefore its ultimate function.
So form follows sequence, which dictates function.
Precisely. And that's why abnormalities in the primary structure can be so devastating. The source material really emphasizes this. A vast majority of known genetic diseases, things like sickle cell anemia, cystic fibrosis, they stem from tiny errors in this primary sequence. Often just a single amino acid change in a chain hundreds long.
Just one wrong brick can compromise the whole building. And the source makes a distinction between polypeptide and protein.
Yeah, it's a bit arbitrary, based on size. Generally, a chain with more than 10 amino acids is called a polypeptide. If it's longer, usually more than 50 amino acids, and typically folded into a functional shape, we tend to call it a protein. But the underlying principle is identical. The sequence is paramount.
Okay, so what actually holds these amino acids together in that specific sequence? What's the glue?
The glue is an incredibly strong covalent bond called the peptide bond. This bond forms specifically between the alpha carboxyl group, a COOH, one amino acid, and the alpha amino group, a methylmin, the next amino acid in the chain.
And water gets released, right? A dehydration reaction.
Yes, exactly. A molecule of water H2O is removed in the process. So you end up with this CONH2 linkage between the two amino acid residues.
And these bonds, they're the real cementing material of the protein world, as the source puts it. They're strong.
Very strong, covalently. But they also have some really important characteristics regarding their geometry. They are rigid and planar.
Cleaner, meaning the atoms involved lie flat.
Yes, the CO group, the NH group, and the two alpha carbons they connect all lie roughly in the same plane. This is because the peptide bond has partial double bond character. Resonance between the CO and CN bonds prevents free rotation around that CN axis.
Ah, okay. So it's not like a freely rotating single bond. That rigidity must limit how the backbone can twist and turn.
Critically important. It significantly limits the possible conformations the polypeptide backbone can adopt, which makes the folding process much more constrained and predictable, contributing to the stability of the final structure.
And they usually exist in a specific configuration.
Yes, the trans configuration is strongly favored. This means the alpha carbons, and thus the R groups, of adjacent amino acids are on opposite sides of the peptide bond. This minimizes steric hindrance, or bumping, between those potentially bulky R groups. The cis form, where they're on the same side, is much rarer, usually only seen with proline.
Okay, trans is standard, and even though the bond itself is strong, parts of it are polar.
That's crucial. The carbonyl oxygen has a partial negative charge, and the amide hydrogen has a partial positive charge. This polarity within the backbone itself is absolutely essential.
Why?
Because these are the very groups that will participate in forming the hydrogen bonds that stabilize the next level of structure, the secondary structure, like alpha helices and beta sheets. The backbone itself provides the H-bond donors and acceptors.
Got it. So the peptide bond provides strength, rigidity, and the H-bonding capability for later folding. And we always read the sequence in one direction.
Yes, the convention is absolutely fixed. The sequence is always read and written, starting from the N-terminal residue, the amino acid at the end, with the free, unbonded alpha amino group. That's considered the beginning or the left side.
N for nitrogen, N for numero uno.
If that helps. And it ends at the C-terminal residue, the amino acid at the other end, with the free, unbonded alpha carboxyl group. That's the finish, or the right side.
N terminus to C terminus, left to right.
And this convention isn't just arbitrary. It actually mirrors the direction in which proteins are synthesized by the ribosome in the cell. Synthesis starts at the N-terminus and proceeds towards the C-terminus.
Okay, so we know the sequence is vital, but how did scientists actually figure out the primary structure of a protein? Especially back in the day before we could just sequence the gene. It sounds incredibly complex.
It was, and still is, a complex undertaking requiring meticulous work. The source outlines a classical three-stage approach, which always starts with getting a highly purified sample of the protein you're interested in.
Right, you need to know you're only sequencing one thing. What's stage one?
Stage 1 is determining the overall amino acid composition. This doesn't tell you the order, just what amino acids are present and in what relative amounts.
How do you do that?
You take your pure protein and completely break it down into its individual amino acids. This requires complete hydrolysis, breaking all the peptide bonds, usually done with strong acid, like 6-MHCl at a high temperature for many hours.
That sounds pretty harsh.
It is, but it gets the job done. Then, you take that mixture of free amino acids and separate them, typically using ion exchange chromatography. As the different amino acids come off the column, you react them with a reagent like 9-hydrin, which makes them colored, and quantify the amount of each one.
Okay, so stage one, know your inventory of bricks. What's stage two?
Stage 2 is fragmentation. Most proteins are way too long to sequence in one go, even with modern methods, so you need to break the long polypeptide chain into smaller, more manageable peptide fragments.
Okay, chop it up. But how do you do that reliably?
Well, first, if your protein is made of multiple polypeptide chains, subunits, held together by non-covalent forces, you need to separate those using denaturing agents like urea or guanidine hydrochloride. And if there are any disulfide bonds holding chains together or forming loops within a chain, those need to be broken too, usually by chemical reduction in alkylation or oxidation with performic acid.
Right, get down to individual linear chains first, then chop.
Then you chop using highly specific methods. Enzymatic cleavage is very common. The enzyme trypsin is a workhorse here. It's a protease that cuts the polypeptide chain very specifically, but only on the carboxyl side of lysine, Lis, K, or arginine, R residues.
So it cuts after K or R, predictable cuts.
Highly predictable. Another enzyme, chymotrypsin, cuts after large hydrophobic residues like phenylalanine, Phe-F, tryptophan, Trep, W, and tyrosine, T or Y. Using different enzymes gives you different sets of fragments.
Are there chemical ways to cut too?
Yes. Chemical cleavage. The classic example mentioned in the source is cyanogen bromide, nathrm-CN-Br. This chemical reagent is unique because it specifically cleaves the peptide bond only on the carboxyl side of methionine, MET-M residues.
After methionine. Okay, so using trypsin gives one set of fragments, CNBR gives another set. Why do you need different sets?
Because that's how you figure out the order. By generating different sets of overlapping fragments, you can piece the full sequence back together like a jigsaw puzzle. That overlap is absolutely key for the final assembly stage.
Ah, okay. Fragmentation gives you puzzle pieces. What's stage 3?
Stage 3 is the actual sequence determination of those individual peptide fragments, figuring out the amino acid order within each small piece.
How's that done?
Historically, the first major breakthrough was Sanger's reagent, which is FDNB, 1-fluoro-2,2-nitrobenzene. Sanger used this to determine the sequence of insulin Nobel Prize-winning work. FDNB reacts specifically with the free N-terminal amino group of a peptide.
So the tag's the first amino acid.
It tags the N-terminal amino acid, but the downside was, to identify which amino acid got tagged, you then had to completely hydrolyze the rest of the peptide chain with acid. So you found out the first amino acid, but you destroyed the rest of the fragment in the process. You'd have to start over with more material to find the second one. Very laborious.
Like finding the first brick, but having to demolish the wall to see it, as the outline said, not ideal.
Not ideal at all. The real revolution came with Edman degradation, developed by Per Edman. What's truly fascinating here is the elegance of his chemistry, using Edman's reagent, which is phenyl isothiocyanate, PITC.
How does that work?
PITC also reacts with the N-terminal amino group, but under mild acidic conditions, it allows you to cleave off only that first amino acid as a derivative, a PTH amino acid, phenylthioidantoine derivative, leaving the rest of the peptide chain completely intact.
Whoa, So you identify the first one, and the rest of the peptide is still there.
Exactly. It allows for sequential degradation. You identify the first PTH amino acid, then you take the remaining peptide, now one residue shorter, and react it again with PITC to identify the new N-terminal amino acid. You could repeat this cycle over and over again.
Peeling off the amino acids one by one from the N-terminus. That's brilliant.
It was a game changer. This process was later automated into a machine called a sequenator. The machine performs the Edman chemistry cycles automatically and identifies each released PTH amino acid as it comes off, usually using high-performance liquid chromatography, HPLC.
Okay, so the sequenator sequences the fragments, but how do you get the full protein sequence?
That's where those overlapping peptides come back in. You sequence all the fragments you got from cutting with trypsin, then you sequence all the fragments you got from cutting with CNBR. Because the cut sites are different, the fragments will overlap.
So a fragment from the trypsin digest might start halfway through a CNBR fragment and end halfway through the next one.
Exactly. By finding these overlaps, you can logically deduce the correct order of all the fragments, and therefore reconstruct the entire original primary sequence of the protein. It's like putting the jigsaw puzzle together using the edges of the pieces.
Okay, we've built the primary chain, the linear sequence, but a straight chain doesn't really do much, does it? The function comes from the folding.
Precisely. The linear sequence contains all the information needed for the protein to spontaneously fold into its unique and functional three-dimensional shape. This folding process happens in a hierarchy of structural levels, starting with secondary structure.
Okay.
This is the first level of actual folding. It refers to the regular, repeating spatial arrangements of adjacent amino acid residues in the polypeptide chain. It's about local folding patterns.
Not the whole protein shape yet, just local segments twisting or arranging themselves.
Exactly. And the entire premise of secondary structure relies on one key stabilizing force: extensive hydrogen bonding. But critically, it's hydrogen bonding between the backbone atoms, the CO and NH groups of the peptide bonds themselves, not involving the R groups at this stage.
Okay, backbone H bonds hold secondary structure together. What are the main types?
The two major, most stable types are the alpha helix and the beta pleated sheet. The beta helix is probably the most common. It's a tightly packed, coiled structure, almost always right-handed, like a standard screw thread.
Like a spiral staircase.
A bit like that, yes. It's stabilized by hydrogen bonds formed within the helix itself. Specifically, the CO group of one amino acid, let's call it residue N, forms an H bond with the NH group of the amino acid that is 4 residues further down the chain, residue N plus 4.
Okay, N to N plus 4 bonding. That sounds like a very regular pattern.
It's extremely regular. This pattern ensures that almost all the peptide bonds, CO and NH groups within the helix, except for the first few NH groups at the start and the last few CO groups at the end, are participating in this hydrogen bonding network. It maximizes stability.
And how many amino acids does it take to make one turn of the helix?
It averages 3.6 amino acid residues per turn. And another key feature is that the R groups, the side chains, project outward from the helical axis.
Sticking out from the spiral, why is that important?
It minimizes steric clashes.
Yeah.
If the R groups were pointed inward, the core would be too crowded, especially with bulky side chains. Pointing outward allows them to interact with the environment or other parts of the protein later on.
Okay. Alpha helix, right-handed coil, 3.6 residues turn, N to N plus 4 H bonds, R groups out. Are there amino acids that don't like being in an alpha helix?
Yes, definitely. We mentioned proline earlier.
The kink inducer.
Right. Because its side chain is bonded back to the backbone nitrogen, proline lacks the NH group needed to donate that crucial hydrogen bond within the helix. Plus, its rigid ring structure doesn't fit the required helical angles. So proline is often called a helix breaker. It disrupts or terminates an alpha helix.
Any others?
Also, having a long stretch of amino acids with the same charge, say, several glutamates or lysines in a row, can be disruptive due to electrostatic repulsion between the R groups. And very bulky R groups close together can sometimes cause steric hindrance.
Okay, so alpha helix is one major type. What's the other?
The other is the beta-pleated sheet. Unlike the coiled helix, beta sheets are formed from polypeptide segments that are almost fully extended. Think of them as stretched-out strands.
Beta strands.
Yes, beta strands.
Mhm.
A beta sheet is composed of 2 or more of these beta strands lying side by side. The stabilization, again, comes from hydrogen bonds between the backbone CO and NH groups.
But if the strands are side by side, the H bonds must form between the strands, right? Not within a single strand like the helix.
Exactly. The H bonds form between adjacent beta strands. These can be strands from different polypeptide chains, interchain H bonds, or more commonly, segments of a single polypeptide chain that fold back on themselves, interchain H bonds.
Okay. And how are the strands arranged relative to each other?
They can be arranged in two ways. In a parallel beta sheet, the adjacent strands run in the same direction, example, both N-terminus to C-terminus. In an anti-parallel beta sheet, the adjacent strands run in opposite directions.
Does that affect the structure?
Slightly. The hydrogen bonding pattern is a bit different, and anti-parallel sheets tend to be slightly more stable. The R groups in a beta sheet project alternately above and below the plane of the sheet, creating that pleated appearance.
Okay, so extended strands, H-bonded side by side, parallel or anti-parallel, R groups alternating up and down. Where do you typically find beta sheets?
They are often found forming the rigid core structure in many larger globular proteins. They provide a strong, stable framework. Think of silk fiber, for instance. Its strength comes largely from extensive anti-parallel beta sheets.
All right, so we've got local coils, alpha helices, and sheets, beta sheets forming in the secondary structure. What happens next? How does the whole chain fold up?
That brings us to the tertiary structure. This level describes the overall, complete three-dimensional arrangement of the entire polypeptide chain, including how those alpha helices and beta sheets, plus any loops or turns connecting them, fold up relative to each other in space.
So this is the final 3D shape of a single polypeptide chain, the functional shape.
Exactly. This is the level where the protein typically achieves its biological activity. It results in a compact, often roughly spherical, globular, or elongated fibrous structure.
And what drives this overall folding? We talked about polarity.
Polarity is huge here, especially for globular proteins folding in water. The formation of the hydrophobic core is arguably the single most important driving force. The polypeptide chain folds spontaneously in such a way as to bury the hydrophobic nonpolar R groups in the interior, minimizing their unfavorable contact with water.
Hiding the oily bits again.
Hiding the oily bits. Simultaneously, the hydrophilic polar and charged R groups are preferentially positioned on the exterior surface, where they can happily interact with the surrounding water molecules or other cellular components.
Makes sense thermodynamically. But once it's folded, what holds that specific tertiary shape in place? It must be more than just hiding from water.
Absolutely. The tertiary structure is stabilized and locked into place by a whole range of interactions, primarily involving the R groups this time. We have both covalent and non-covalent forces at play.
Hey, what's the covalent one? There's only one main type, right?
The only covalent bond that typically contributes to stabilizing tertiary and sometimes quaternary structure is the disulfide bond.
Formed between two cysteines.
Yes, formed by the oxidation of the sulfhydrylyl groups of two cysteine residues, creating a strong covalent link between different parts of the polypeptide chain, or sometimes between different chains. These disulfide bonds are like permanent structural staples.
And you find them more often in proteins that exist outside the cell.
Very often, yes. Proteins that are secreted from the cell, like antibodies or digestive enzymes or hormones like insulin, often rely heavily on disulfide bonds for stability, because the extracellular environment can be much harsher, more oxidizing variable pH than the inside of the cell.
Okay, disulfide bonds are the covalent glue. What about the non-covalent forces? These must be important too.
Hugely important. In fact, for most proteins, especially intracellular ones, the tertiary structure is primarily maintained by the cumulative effect of a vast number of relatively weak non-covalent interactions.
What kinds?
There are several key types. Hydrogen bonds can form between polar R groups, like serine, aterine, asparagine, glutamine, or between polar R groups and backbone atoms, or between R groups and water. Ionic interactions, also called salt bridges, occur between positively charged basic R groups, all ice, arg, his, and negatively charged acidic R groups, asp, glue.
The positive-negative attraction.
Exactly. Then, of course, there are the hydrophobic interactions, the tendency of nonpolar R groups to cluster together away from water, which we said is a major driving force for folding itself. And finally, there are the very weak, short-range van der Waals forces, which are attractive forces that occur between any two atoms that are very close together.
So it's the sum total of all these weaker forces, H bonds, ionic bonds, hydrophobic packing, van der Waals, plus maybe some disulfide staples that holds the whole 3D structure together.
Precisely. It's a collective effort. Lots and lots of weak interactions add up to create a stable, specific, tertiary structure.
And sometimes within that overall tertiary structure, there are distinct regions, domains.
Yes, that's a very important concept, especially for larger proteins. Domains are distinct, structurally stable, and often functionally independent regions within a single polypeptide chain. A large protein might consist of two or more domains, each perhaps responsible for a different aspect of the protein's function, like binding a substrate or anchoring to a membrane. Domains often fold independently of each other.
Okay, so tertiary structure is the final shape of one polypeptide chain. But some proteins are made of more than one chain, aren't they?
Yes, many are. And that brings us to the highest level of protein organization, quaternary structure. This level only applies to proteins that are composed of two or more separate polypeptide chains, which are often called subunits.
So if a protein is just a single chain, it only has primary, secondary, and tertiary structure. But if it's made of multiple chains associating together, that association is the quaternary structure.
Exactly. Such proteins are called oligomers, or multimeric proteins. A protein with two subunits is a dimer, three is a trimer, four is a tetramer, and so on. Hemoglobin, the oxygen carrier in our blood, is a classic example of a tetramer. It has four subunits, two alpha chains and two beta chains.
Okay. And what holds these separate subunits together in the final complex? Is it more peptide bonds or disulfide bonds?
No, and this is a critical point. The subunits in a quaternary structure are held together exclusively by non-covalent interactions.
Only non-covalent. So the same forces that stabilize tertiary structure, but now acting between different polypeptide chains.
Precisely the same forces: hydrogen bonds, ionic interactions, salt bridges, hydrophobic interactions, often very important at the interface between subunits, and van der Waals forces. There are generally no covalent bonds linking the subunits together,
Why is that significant? Why rely only on weaker forces to hold the whole complex together?
because it allows for flexibility and regulation. The fact that subunits are held by weaker non-covalent bonds means their relative positions and interactions can change subtly. This is the basis for allosteric regulation, which is incredibly important for controlling metabolic pathways and cellular functions.
Allosteric meaning action at a distance.
Sort of. It means that the binding of a molecule, like a substrate, activator, or inhibitor, to one site on one subunit can trigger a conformational change, a shape shift, that is transmitted through the quaternary structure to affect the activity or binding properties of the other subunits.
Like in hemoglobin, binding oxygen to one subunit makes the others bind to oxygen more easily.
Exactly. That's cooperative binding, a classic example of allostery mediated by quaternary structure changes. The relatively weak non-covalent bonds between subunits allow for this essential communication and functional integration between the different parts of the protein machine. It allows for much more sophisticated regulation than a single rigid polypeptide chain could achieve.
Okay, we've assembled the protein from primary sequence right up to potentially complex quaternary structures. Now let's think about how these finished molecules actually behave, say, in a test tube or in the cell. What are some of their key physical properties? Hashtag 4.1 physical properties and colloidal nature?
Well, the most obvious property is their sheer size. Proteins are macromolecules, meaning they are very large compared to simple molecules like water or glucose. Their molecular weight varies enormously depending on the number of amino acid residues they contain.
What's the typical range?
The average molecular weight of a single amino acid residue is about 110 daltons. So a small protein like insulin, which has 51 amino acids, has a molecular weight around 5,700 Day. A medium-sized protein like hemoglobin, with about 574 residues in its four chains, is around 64,450 Day. And some protein complexes involved in muscle structure or DNA replication can have molecular weights in the hundreds of thousands or even millions of daltons.
So huge variation. And because they're so large, they don't really dissolve in water like salt or sugar, do they?
That's right. Due to their immense size, proteins don't form true solutions in water. Instead, they form colloidal solutions, or colloidal dispersions. The large protein molecules remain dispersed and suspended in the water, usually due to hydration layers and surface charges, and they don't settle out under gravity.
But they're too big to pass through certain membranes.
Exactly. They typically cannot pass through semi-permeable membranes, like dialysis tubing, which have pores large enough for water and small solitudes, but too small for large protein molecules. This property is widely used in biochemistry for separating proteins from smaller molecules, a process called dialysis.
Okay, colloidal nature is important. How else do we classify proteins based on their properties? The source mentioned solubility and composition.
Yes, there are broad classifications based on chemical nature and solubility. The simplest category is simple proteins. Upon hydrolysis, these yield only amino acid residues and nothing else.
Examples.
Albumins, like serum albumin in blood, soluble in water, and globulins, like antibodies, often soluble in dilute salt solutions, but not pure water, are common examples of globular simple proteins. Fibrous simple proteins include things like keratins, insoluble structural, and collagens, insoluble structural.
Okay, simple means only amino acids. What's the next type?
Conjugated proteins. These are proteins that, upon hydrolysis, yield not only amino acids, but also some non-protein component, which is called a prosthetic group. This prosthetic group is essential for the protein's biological activity.
So it's a protein plus something else attached, like what?
Lots of possibilities.
Yeah.
Nucleoproteins have nucleic acid, DNA or RNA attached, like ribosomes or chromosomes. Glycoproteins have carbohydrate chains attached, common for cell surface receptors and secreted proteins. Lipoproteins have lipids attached, crucial for transporting fats in the blood. Chromoproteins have a colored pigment as their prosthetic group.
Like hemoglobin, its prosthetic group is the iron-containing heme group, which gives blood its red color.
Exactly. The heme group is essential for oxygen binding. Without it, the globin protein part is non-functional for oxygen transport.
Okay, simple and conjugated. Is there a third category?
The source also mentions derived proteins. This isn't really a structural class, but rather refers to products formed when native proteins are altered by heat, enzymes, or chemical agents. It includes denatured proteins or smaller peptide fragments produced by partial hydrolysis.
All right, so proteins are big, colloidal, and can be simple or conjugated. But maybe the most defining physical property, the one that really dictates how a protein behaves in solution, especially in response to pH changes, is its isoelectric pH, or PI. So what does this all mean?
The PI is absolutely fundamental. It's defined as the specific pH value at which the protein molecule exists as a Switterian and carries zero net electrical charge.
Zero net charge, meaning the total number of positive charges on the protein exactly balances the total number of negative charges.
Precisely. Remember, proteins have many acidic R groups, asp, glue, C terminus, that can be negatively charged, and many basic R groups, mol, Is, arg, his, N terminus, that can be positively charged, mothrum, NH3, or similar. The PI is the unique pH, where the sum of all positive charges equals the sum of all negative charges.
And the value of the PI must depend heavily on the amino acid composition.
Absolutely. A protein with a high proportion of acidic amino acids, asp, glue, will have a low PI, meaning it needs a very acidic environment, low pH, to protonate enough groups to reach neutrality. Conversely, a protein rich in basic amino acids, Lys, arg, his, will have a high PI, requiring an alkaline environment, high pH, to deprotonate enough groups to become neutral.
Can you give some examples?
Sure. Pepsin, the digestive enzyme that works in the highly acidic environment of the stomach, pH 1 and 2, has a very low PI, around 1.1. Casein, the main protein in milk, has a PI of 4.6. Human serum albumin is around 4.9. Lysozyme, an enzyme found in tears and egg white that has many basic residues, has a high PI of about 11.0.
Wow, huge range from 1.1 to 11.0. Okay, so the PI is the pH of net neutrality. Why is this so important practically?
Because at its PI, a protein exhibits several key properties. Since it has no net charge, the repulsive forces between protein molecules are minimized, and the attractive forces can dominate. Also, its interaction with water is often weakest at the PI. Therefore, at its isoelectric pH, a protein typically exhibits minimum solubility,
Least soluble when neutral.
and consequently, it exhibits maximum precipitability. Proteins are most likely to aggregate and precipitate out of solution when the pH of the solution is adjusted to equal their PI.
Ah, so you can selectively precipitate a protein out of a mixture by adjusting the pH to its specific PI.
Exactly. It's a classical method used in protein purification. Find the PI, adjust the pH, and your protein of interest might crash out of solution while others remain dissolved. Also at the PI, the protein has its least buffering capacity. It's least able to resist changes in pH right at that neutral point.
And the source gave that great example of milk souring.
Yes, the flocculation of milk. Milk is a colloidal suspension of casein proteins, PI 4.6, and other components. When milk sours, bacteria produce lactic acid. This lowers the pH of the milk. As the pH approaches 4.6,
The PI of casein.
the casein molecules lose their net negative charge, which normally keeps them repelling each other and dispersed. They become neutral, their solubility plummets, they start to aggregate together, and they precipitate out.
Causing the milk to curdle or flocculate, a direct consequence of hitting the isoelectric point. Very clear.
So we've seen proteins have specific structures essential for function and properties like PI that govern their behavior. But these structures, especially the tertiary and quaternary ones held by weak forces, are quite delicate. And that leads us to the concept of denaturation.
Denaturation, that sounds bad. Like undoing all that careful folding.
It is essentially the disorganization of the native biologically active protein structure. Denaturation involves the disruption and loss of the secondary, tertiary, and, if present, quaternary levels of structure.
So the helices unwind, the sheets fall apart, the overall 3D shape is lost, and subunits might separate.
Exactly. But here's the crucial point to grasp. Denaturation typically does not involve the breaking of the strong covalent peptide bonds. The primary structure remains intact.
So the sequence of amino acids is still the same, but the way it's folded is completely messed up.
Precisely. Denaturation is primarily about breaking the weaker interactions that hold the folded structure together. The hydrogen bonds, the hydrophobic interactions, the ionic bonds, and sometimes it also involves breaking the covalent disulfide bonds if they are present.
And what are the consequences of losing that specific 3D shape?
The primary consequence is a loss of biological activity. Protein function is exquisitely dependent on its precise shape. Think of an enzyme's active site or an antibody's binding site. If you disrupt the shape, you destroy the function.
No shape, no job.
Pretty much. Other consequences often include increased viscosity as the unfolded chains take up more space, and decreased solubility, often leading to precipitation as hydrophobic groups become exposed. Think about cooking an egg white.
It goes from clear and runny to white and solid.
Right. The heat denatures the albumin proteins.
Yeah.
They unfold, expose hydrophobic regions, aggregate together, and precipitate, forming that solid white mass. The primary structure is still there, but the functional soluble protein is gone.
And that cooking example highlights one cause of denaturation: heat. What else can do it?
Many things can act as denaturing agents. Physical agents include heat, as we said, but also violent mechanical agitation or shaking, like whipping egg whites into meringue, exposure to X-rays, or UV radiation.
Okay. Physical stresses. What about chemicals?
Chemical agents are also very effective. Extremes of pH-strong acids or strong alkalis disrupt ionic bonds and hydrogen bonds. Organic solvents like alcohol or acetone disrupt hydrophobic interactions. That's why alcohol swabs are used as disinfectants. They denature bacterial proteins. High concentrations of urea or guanidine hydrochloride are classic lab reagents used to denature proteins by disrupting H-bonds.
And heavy metals.
Yes, salts of heavy metals like lead or mercury, math or Hg2+1, are potent denaturants. The positive metal ions react strongly with negatively charged R groups and often with sulfhydryl groups, disrupting salt bridges into sulfide bonds, causing the protein to precipitate.
And the source mentioned using raw egg white for mercury poisoning based on this.
It's an old traditional remedy, the idea being that the abundant albumin protein in the egg white will hopefully react with and precipitate the ingested mercury ions in the stomach before they can be absorbed into the bloodstream. The denatured protein metal complex might then pass through the digestive system. It's not a guaranteed cure by any means, but it's based on that principle of denaturation by heavy metals.
Interesting. Now, is denaturation always permanent? Once you fry an egg, it stays fried?
Usually it is irreversible, especially if caused by harsh conditions like strong heat or chemicals. The unfolded protein chains often aggregate in ways that prevent them from refolding correctly, even if the denaturing agent is removed. However,
There's a but.
there is. Sometimes if the denaturation is carried out gently and the denaturing agent can be completely removed carefully, some proteins can spontaneously refold back into their native active conformation. This process is called renaturation.
So the primary sequence really does contain all the information needed.
It seems so, at least for some proteins under the right conditions. The source mentions hemoglobin denatured by salicylate, like aspirin, can sometimes renature. It highlights that while the weak forces are easily disrupted, the underlying primary sequence holds the ultimate blueprint for the correct fold. #outro. So looking back, we've really traced the complete protein story today. We started with the surprisingly simple chemistry of just 20 unique alpha-amino acid bricks, each with its own personality defined by its R group.
Yeah, then we saw how they're linked by strong peptide bonds into a specific primary sequence, the absolute blueprint dictated by our genes.
And how that sequence then guides the local folding into secondary structures like helices and sheets, stabilized mainly by backbone hydrogen bonds.
Which then collapse and arrange themselves into the functional tertiary structure, driven by hydrophobic forces and locked in place by a combination of R group interactions, H bonds, ionic bonds, van der Waals, and sometimes those disulfide staples.
And finally, for some proteins, how multiple folded subunits come together via non-covalent forces to form the complex quaternary building, allowing for sophisticated regulation. It's quite a journey from single amino acid to functional machine.
It really is. And the insight that keeps hitting me is the profound elegance, but also the seeming fragility of it all, especially at that quaternary level. The stability and the regulatory power of these massive, essential complexes depend almost entirely on the collective strength of those relatively weak, non-covalent forces.
Hydrogen bonds, hydrophobic interactions, electrostatics.
Right. It's like achieving functional genius through a massive committee of weak interactions working together. It's kind of amazing it holds together so well.
It is, but it also raises an important question, doesn't it? We focus so heavily today on the roles of the standard 20 amino acids in building these large protein structures and acting as catalysts.
Right, the main construction job.
But we also had that fascinating detour into the world of non-standard and non-protein amino acids, and also biologically active peptides, short chains of amino acids. The source mentioned things like the nonapeptides oxytocin, involved in childbirth and bonding, and vasopressin, regulating water balance, and of course the neurotransmitter GABA, derived from glutamate.
Yeah, small molecules but powerful functions, separate from being part of a big protein.
Exactly. So if the standard 20 amino acids are the primary construction crew building the big cellular machinery, what other specialized, maybe more subtle, non-protein roles are these foundational components, or slight variations of them, playing in managing things like cellular signaling, neurological function, maybe even disease prevention or progression?
Are we only just scratching the surface of the full chemical versatility that these simple building blocks offer beyond just making proteins?
It feels like we might be. It's definitely something for you to mull over, think about the sheer diversity of function packed into these fundamental molecules, the very architecture that keeps you and every other living thing running.